Owing to mutation, the genetic makeup of a cell population can display diversity instead of a pure genotype characteristic of a well-defined species. In these situations, quasi-species models are used to think about genotypic distributions. This slide deck outlines how eigenvalue-eigenvector analysis can be used to study the green box in the paper Quasi-Species Made Simple by authors Bull, Myers, and Lachman in PLOS Computational Biology from 2005. We divide this slide deck into two videos. In this video, we first describe the dynamics of the model system. In the next video, we outline the solution to the model in terms of eigenvalues and eigenvectors. Mutation can generate a variety of alleles from an initially genetically homogeneous population. This model tracks the populations of cells with either of two alleles. At an initial time, there are N1 cells with allele A1 and N2 cells with allele A2. What are the populations in the future after a duration delta t has elapsed? In order to calculate the dynamics of the populations, we specify microscopic dynamics at the level of the single cell. Consider this cell with allele 1. By the time delta t has elapsed, it might have generated progeny. When we say that cells with allele 1 can replicate, we mean that for each cell of A1 at time t, we expect to have W1 cells at time t plus delta t. Here, the number W1 is 3. Note, however, that not all of the W1 of these resulting cells are blue. Owing to mutation, some cells might be orange, having allele 2 instead of allele 1. Thus, we subtract from the W1 cells at time t plus delta t a fraction mu sub 1 that is orange, and these cells actually become part of the population of cells with allele 2 at time t plus delta t. We've been looking at progeny and mutants resulting from a single cell. Our table is quantified in terms of a single cell. Suppose that each of the cells in the cell population N1 performs these same processes according to these same parameters. Then the contribution to the population N1 at later time t plus delta t, owing to the faithful survival and reproduction originating in the population N1 at earlier time t, is N1 of t, the number of cells initially available to do something, multiplied by W1 times the factor 1 minus mu1, the net effectiveness of cells of type 1 on a per-cell basis of being able to survive and reproduce without mutation. In the same way, the portion of the population of cells with allele A2 at time t plus delta t, owing to survival and reproduction with mutation of cells initially having allele A1, is the size of the source population N1 of t multiplied by W1 mu1. Consider this highlighted orange cell. It can also survive and reproduce. We say that A2 replicates, meaning that for a given A2 at time t, we expect to have W2 cells at a future time t plus delta t. Owing to mutation, some of these W2 cells have allele A1. So for each orange cell at time t, we expect to have W2 times the mutation fraction mu3 blue cells at a later time t plus delta t. Some of the W2 cells at time t plus delta t corresponding to an initial orange cell at time t might now be mutants that have another allele, for example, some sort of allele A3 not explicitly tracked in this model. For simplicity, assume that these mutations are irreversible, so that any cellular population lost to allele A3, and so on, never returns either to A2 or A1. These mutants might as well be considered dead. We use mu2 to denote the fraction of the W2 cells that are mutants, either mutants with allele A1 or mutants with another allele neglected by this model. The fraction of the W2 cells that has an allele other than A1 or A2 at time t plus delta t is mu2 minus mu3. We have been highlighting the dynamics associated with one of the initially orange cells. Let's say that each of the initially orange cells behaves in the same way. Then a contribution to the orange population N2 of t plus delta t can be calculated by multiplying the number of orange cells initially available to do something that's N2 of T, by their effectiveness in contributing to the orange population, W2 times the quantity 1 minus mu2. Orange cells are not as effective at contributing to their own future populations if the mutation fraction mu2 is high. A contribution to the blue population N2 of T plus delta T can be identified as the product of the population of orange cells available to do something at time t, multiplied by their effectiveness, W2 mu3, in generating blue cells. You can do finger exercises to confirm that dynamics of the system can be expressed using matrices. Using a flowchart like the one at the top of this page, the authors summarize the microscopic model that we've just animated. The heights of the blue and orange bars indicate that the authors studied the particular example in which W1 exceeded W2. An initial blue cell would correspond at time t plus delta t to more cells than would be expected to originate from an initial orange cell. The authors call the W's the fitnesses of blue and orange cells, so in this sense, blue is more fit than orange. In addition to faithful survival and reproduction, blue cells can turn orange, and orange cells can turn either blue or some other color not explicitly tracked by the model. Now that we have described the model, we solve for its dynamics in the next video.